So it's great to be back at Berkeley, and I want to thank Laurent and the other organizers for the chance to come back and kind of revisit old haunts and tell you what we've done in the interim period since I've left. I've been back for a day here, and we've had fog already, the curious smells surrounding the BART station, and I feel like if we have an earthquake, I'll hit the trifecta and revisit everything that I want out of a California experience. So today I'm going to tell you a story about work that's come out of our lab just in the last year or two, and that's caused us to rethink how viruses engage the host and what we think of a canonical antiviral response. And it's going to build a lot on work that Stacy just talked about, and centering around this theme of continual epidemics. So as we all know, in 2009, a new virus emerged in Mexico, and this resulted in the 2009 pandemic. And I love to show this movie because this captures only 4 weeks of that outbreak. And that virus starts in Mexico and very rapidly over the course of these 4 weeks becomes a global pandemic. And it's a really powerful demonstration of how quickly and broadly these viruses can move. And if you overlay air travel, you see this beautiful correlation between virus transmission and where people are flying and moving. And this was the 2009 pandemic. It moved very quickly, but we dodged a bit of a bullet in that at the end of the day, while there were around 90 million infections, the mortality rate was on par, if not below, seasonal influenza virus. So we had a highly transmissible virus, but very low pathogenicity. And this contrasts pretty much with high-path viruses. So Stacy alluded to these. These are viruses that are highly pathogenic. They're transmitting directly from birds into people. And while they don't occur very frequently, so since 1997, there have only been about 850, 900 cases that have been identified as H5N1. If you do get this, it's bad. You've got about a 60% case fatality rate. And more concerning from H5 is actually the H7 and 9. So these have been emerging in China over the last few flu seasons, and these are moving directly again from birds into people. And they also have high case fatality rates on the order of 30%. So things that we get really concerned about then is what should happen if you had the transmissibility of a 2009 pandemic virus and just a tiny touch of the pathogenicity of these high-path avian viruses. And as Stacy noted, it's the 100-year anniversary of that exact event. So in 1918, we had that emergence of a highly pathogenic virus in humans that killed about 50 million people. So why, 100 years on from that pandemic, are we still thinking and worried about flu? And in fact, flu's been in the lab as an isolated clone since 1933. Why are we still here today? One of the major concerns and one of the major problems with this disease is that it exists in a stable reservoir. The reservoir are migratory waterfowl, and then they transmit viruses into domesticated chickens, ducks, turkeys. And in these environments, the virus is largely asymptomatic, it's not terribly pathogenic, and you have this huge diversity of different variants of virus circulating in this stable reservoir. And it's this spillover then into other hosts that continually repopulates disease in humans. And that's why we have this constant epidemic again and again and again, year after year after year. Even though we develop population level immunity, it's these new viruses that are spilling out that result in transmission into intermediate species such as pigs and then jumping over into people. This is exactly the case of the 2009 virus. We know that that virus was actually a reassortant containing genes from both avian, swine, and humans, and those viruses emerged from pigs into people. This annual epidemic results in about 50 million infections worldwide and over $30 billion in cost in the United States alone. And as we've heard, this year in particular is a very bad year, with H3N2 dominating. And I pulled headlines off of the Canonical and the failing New York Times to highlight the importance of these disease outbreaks and the fact that we get complacent as a society thinking about influenza and thinking about a vaccine and so on. But really, there's a lot of work yet to be done, and that it's still a continual public health problem. So where does my lab go, and what do we think about, and how do we try to address these problems? Or to put it in another way, when we're asking for money, why aren't we all dead? Why hasn't flu killed us? And it's because while viruses are circulating in a stable population in the avian hosts, they can't simply just move over into people. They actually face large barriers to cross-species transmission. And these barriers can be broken down at the molecular level, and that's really where my lab focuses. We try to understand the molecular virology of influenza virus and how that impacts host range and pathogenicity. In the context of cross-species transmission for flu, the barriers come down to the availability of the appropriate receptor and the appropriate host factors. And on the virus side, that really shakes out at the viral entry protein hemagglutinin and the viral replication complex, and in particular, the viral polymerase. And viruses must undergo specific adaptations in these two viral genes to allow them to efficiently transmit from an avian reservoir into mammalian hosts and then transmit within that mammalian host as well. So looking in a little bit more detail then at influenza virus, there are 4 different types of influenza virus, and we focus on influenza A and influenza B because they are the dominant strains infecting humans. On the surface, we have neuraminidase and hemagglutinin. They're both involved in the attachment of the virus to get in and then getting out. And there are 18 different subtypes of hemagglutinin, 11 different subtypes of neuraminidase. In humans right now, we only have 2 groups circulating, H3N2 and H1N1. In addition to influenza A with its various HAs and NAs, we also have 2 major lineages of influenza B. So when you get your vaccine, you actually have 4 viruses in there, an H3N2, an H1N1, and 2 influenza Bs. Flu is made up of 8 genomic segments, and these segments are RNA, and they're in the minus sense. So what that means is they need to be transcribed into plus sense messenger RNA before you can make protein. And as a consequence, these single-stranded RNAs get packaged into ribonuclear protein particles that have the viral polymerase on them. So the virion itself carries the polymerase. And the first thing it does after it gets into a cell is it starts making viral mRNAs. So virions attach to cells using sialic acid receptors. They are then N-acetosed, and upon acidification, you get fusion of the viral and host membranes and deposition of these genomic segments into the cytoplasm of the cell. Now, unusual for an RNA virus, it actually replicates in the nucleus. So the genomic RNAs are imported into the nucleus of the infected cell, where they undergo that pioneering round of transcription. After transcription, these viral messages, some of which are spliced, go out into the cytoplasm to make viral proteins. Those viral proteins are then relocalized to appropriate sites for replication, some of them going onto the membrane and some of them feeding back into the nucleus. So now you can replicate your viral genomes and export these large ribonuclear protein complexes out into the cytoplasm again, where they move off to the membrane, sites of assembly, and then finally budding, and we've got viruses that are left out to repeat this infectious cycle. This is what an infection really looks like. So I've showed you that simplified diagram where we can track all the viral proteins, but in the context of really what's happening, there's this massive interface between the virus and the host, and this is more of an engineering diagram that was put together by Yoshi Kawaoka's lab. And it's not up here for you to actually read any of it, it's just really up here as a demonstration of the complexity of the interactions that are going on. So every box represents a cellular protein or process that's ongoing, and all of the lines represent interactions between viral and host cofactors that are necessary for each step in the virus life cycle. And kind of faced with this overwhelming complexity, how can we get a handle on what these interactions are really doing, and why do we even care? And the reason we care is because each one of these dictates the efficiency of virus replication and the ultimate pathogenicity and outcome of that disease. So we are not the only ones to think about this. There are many groups that have tried to tackle and chip away at each of the different events that are going on here. And what it really comes down to is there are over 3,000 host factors that have been implicated in virus replication. I'm going to tell you the story of one. And it really came from our idea of how could we tackle this problem in a new way to identify functionally relevant and important host factors. And it was driven by a graduate student, V. Tran, and has now been picked up by another graduate student, Mitch Ledwith. And our collaborators in this have been Jocko Boone, Mike Diamond, Martin Schwemmle, and Adolfo Garcia Sastre. So how do people identify host factors important for flu? And what did we learn from that? And that falls into two major camps. There's a functional genomics strategy, and this involves things like RNAi or knockout screening to use genetics to drive your understanding. And then there's more of a proteomic style of things where we've used yeast-2 hybrid, affinity purification, mass spec, or complementation to identify physical interactions. And there are pros and cons of both styles, but really what it comes down to at the end of the day is if you assume there are about 20,000 human genes, over 3,000 of them now have been implicated in influenza virus replication. But of those 3,000, only a few hundred have actually been validated. And of those few hundred, probably only in a handful, maybe a few dozen, have really well-defined molecular mechanisms by which they impact influenza virus replication. And important to us, most of these screens, by the way that they were designed, showed bottlenecks, and the host factors that they identified were biased towards the entry process. And in part, that's because these were survival-based screens. The best way to allow a cell to survive an infection is to never let it get infected. But I've told you my lab is interested in the polymerase and the replication complex, so entry is not important. So how could we look at post-entry events? And the way we could do that is we could get away from influenza virus entry. So influenza virus uses hemagglutinin to enter into host cells, and that's one of the reasons the previous screens picked up all these host factors for hemagglutinin. We used a Frankenvirus. So this is influenza virus, but now it's stably encoding the envelope protein from vesicular stomatitis virus, or VSVG. So VSVG gets into everything, and it uses multiple and redundant pathways, so a single gene knockout won't block entry. So we can bypass HA-mediated entry to get into a cell. Okay, so now we've got a way to get around entry. How could we do a screen? What genetic approach could we take that would allow us to do genome-wide knockouts? So there's this amazing thing I want to tell you about. It's called CRISPR. So we, like everyone else, did a CRISPR screen. And it turns out CRISPR works, right? It's great. So we developed this CRISPR screen where we took a genome-wide knockout library and we knocked out genes of the entire human genome, in theory, in human lung cells, and took this large pool of cells and challenged them with our Frankenstein virus. We then selected cells that survived the infection, presumably because they were missing important host factors. We then re-challenged these, again with that Frankenstein virus, or we counter-screened with bona fide flu, because we wanted to make sure that whatever we found had nothing to do with how we got in, but really had to do about how the virus replicated. We did this screen in duplicate, and at the end of the day, we then isolated DNA from each of these populations, sequenced them to identify the genes that had been knocked out, and then used an algorithm to identify those genes that were knocked out, preferentially waiting for those that are enriched successively across each of these selective events. And when we did that, independent of whether or not we screened using our Frankenstein virus all the way through, or if we counter-screened using bona fide flu, what we ended up with were our top hits for proteins called IFIT2 and IFIT3, suggesting that cells lacking IFIT2 and IFIT3 were somehow less susceptible to influenza virus infection or replication or cell death or whatever the case might be. And what are these IFIT proteins? They are interferon-induced proteins with tetratricopeptide repeats. In humans, they're a family of 4 different proteins. It should be noted right away that these proteins are under a high degree of positive selection and diversification. So when you look at IFITs in humans are similar to IFITs in mice, but they're also quite different and show only about 50% identity, showing rapid evolution at this locus. For IFIT2, our top hit, there's not much known about it. It's massively upregulated during infections and during interferon treatment, and many people use IFIT2 as a positive control to show that interferon induction is occurring. But the molecular function of IFIT2 is poorly defined. It's thought to bind RNA and possibly bind AU-rich elements, so the data on that are fairly weak. We know that it binds other proteins, in particular it homodimerizes with itself shown in the crystal structure here, and it heterodimerizes with IFIT1 and IFIT3. And then high levels of IFIT2 will also induce apoptosis. But the best known function of the IFIT proteins are that they are well-characterized antiviral effectors. So IFIT1 and IFIT2 are known to block replication of a host of different RNA viruses and even DNA viruses. And this is the point where you should be scratching your head and questioning why I'm up here telling you this story, because how did we do a screen for proviral factors and find antiviral effectors? That's also the time when I went back to my graduate student V and I said, what the hell did we just do? We wasted a lot of time and money, and how did we get these results? And the first thing we did then was we wanted to validate the results of the screen. We reached out to Mike Diamond and Jaco Boone, and they shared some knockout cells from mice. And we can see that in these knockout cells and wild-type cells, influenza virus replicates quite well. But now when we lose IFIT2, that virus replication is down by about a log or 2. While we were using the knockout cells from mice, we made our own knockout cells in the human lung cells, and these are human A549 cells. We could knock out IFIT2 in 2 different clonal lines that we generated. And again, influenza virus replication now is suppressed in these clonal populations. So what could we learn, and what is our next question? We know that IFIT2 from humans and IFIT2 from mice are playing this conserved proviral role during the course of infection. And I told you we know a little bit about how the IFITs function, so we started to dissect the known activities of IFIT2 and see if they played a role during the course of influenza virus replication. And our first clue came from the observation that cells lacking IFIT2 don't die. So the normal outcome of an influenza virus infection is cell death. These are our knockout cells and our wild-type MEFs. And when you infect MEFs with influenza virus, be it our normal influenza or our pseudotype virus, it'll clear the well. The cells die very rapidly. But now when you're missing IFIT2, even though the virus is replicating in these cells, they survive the infection, and you let them go 8-10 days out, and they're still doing fine. And this is true not only for our knockout MEFs, but it's also true when we used our knockout human cells, both our knockout human IFIT2 cells that I showed you, and we made knockout IFIT3s. Again, these cells survive challenge, so our control cells die during infection, but we see survival of infection both for a wild type, lab strain, and for our pseudotype. But it's not that IFIT2 knockouts make cells resistant to infection and cell death. That's not the case, because if we took vesicular stomatitis virus and infected these cells, they had no problem dying. And it's not just a lab strain phenomena either. We could see the same thing to varying degrees with primary isolates of influenza B, and then that pandemic 2009 I talked about earlier, and 2 H3N2 isolates. So something about lacking IFIT2 means less virus and less virally mediated cell death. Now, IFIT2 plays a role during apoptosis. Could that be involved in what we're observing here? And we know that IFIT2 induces apoptosis through Bax and BAC. So if this is really the case, a Bax-BAC knockout should phenocopy an IFIT2 knockout. And luckily for us, there are knockout cells from mice where they lack both Bax and BAC, so we can now do those infections again and ask what the consequence was. And sure enough, in cells lacking IFIT2, we once again see this lower level of virus replication, and now cells lacking BAC and BAX also show decreased levels of influenza replication. So we typically think of apoptosis as kind of a last-ditch effort to prevent yourself from spreading virus to your neighboring cells. But now what we're seeing here is, again, a little bit the opposite. That in the absence of apoptosis, when these cells survive infections, there's less virus replication. Could we go after that and kind of test that in a more direct fashion? So we established this experiment where we could initiate an infection with influenza virus and then treat with a caspase inhibitor to block apoptosis, and then measure the viral yield during that 8-hour window. And if we treat cells with caspase inhibitors, sure enough, caspase activity decreases. And now if we measure viral yield coming off of those cells during that same time, we also again see a dose-dependent decrease in the amount of virus that's getting out. So while apoptosis is a last-ditch effort, it appears that it's also important during the context of flu infection. And this helped coalesce for us a couple of papers that have been circulating for the last 10 or so years in the flu field, showing that apoptosis is actually important. And we didn't do any more of the work on it, but what other people have shown is that during apoptosis, during viral infection, you get activation of caspases, and some of those caspase targets are host proteins, and in particular a host protein in the nuclear pore. And you need to cleave that nuclear pore protein to enlarge the pore and let those replicated genomes get back out. And I think that's what we're seeing here, that in the absence of apoptosis due to virally mediated infection, we're not getting efficient cleavage and activation of caspases, and we're getting stuck in the nucleus. So that was pretty satisfying for us, but we knew early on in these experiments that that wasn't the whole story. And the reason we knew that is that we could take those same back knockout cells and our IFIT2 knockout cells and use the reporter viruses we generated to look at really early time points post-infection. And we could see that even beginning at very early time points, there's this divergence between our IFIT2 knockouts, our back knockouts, and our wild-type cells, and that there's a defect in viral gene expression in the absence of IFIT2. And all of this is occurring right when IFIT2 just starts coming up and well before IFIT2 could induce apoptosis. So what's happening at these early stages of infection that's driving this increase in viral gene expression? We showed that this was true not again just for our lab strain, but for many of the viruses we had in the lab. We could look at very early time points at 2 and 4 hours post-infection and see defects in viral gene expression. And this happened in the mouse cells, and it also happened in the knockouts that we created in human cells. So something about IFIT2 was important for viral gene expression. We could complement this defect, right? That's the problem with genetic approaches. You always need to be able to show that the thing that you think you've knocked out is really causing the phenotype. So we could complement the defect in our knockout cells by adding IFIT2 back. So in the absence of IFIT2, we have this defect in viral gene expression, and adding wild-type IFIT2 causes an increase. And we got a first hint as to what might be important. We took an RNA binding of IFIT2 and showed that that mutant no longer complements viral gene expression. So IFIT2 enhances viral gene expression in an RNA-dependent fashion. So what, in the context of an infection then, could be this RNA or the complex in which IFIT2 is interacting? So I told you already that the viral genome is packaged in this large ribonucleoprotein complex that contains the viral polymerase, the genomic RNA, and oligomeric forms of nucleoprotein. And it's this complex from which we transcribe messenger RNAs. So is IFIT2 associating with this ribonucleoprotein complex? So we can infect cells, immunopurify these RNPs, and look for co-precipitating IFIT2. And the answer to that question was no. When we purify RNPs, we don't see any IFIT2. But nucleoprotein exists both in these ribonucleoprotein complexes, and nucleoprotein exists in these free kind of, I don't want to say monomeric states, but it's not part of a replication complex. When we immunoprecipitate nucleoprotein, now we see that nucleoprotein and IFIT2 are associated with each other. These are both RNA binding proteins. So an obvious and immediate question was, is RNA involved in the interaction between IFIT2 and nucleoprotein? And we treated these immunoprecipitates with RNase, and now we could ablate these interactions or disrupt the interactions. So we've got an RNA-dependent interaction between IFIT2 and nucleoprotein. What RNAs are there? Well, we know already that IFIT2 induces viral gene expression, or enhances viral gene expression, and that's dependent upon RNA binding. So are RNAs from influenza being bound by IFIT2? This just confirms that we can get interactions between IFIT2 and NP in an RNA-dependent fashion. So to ask whether or not IFIT2 is binding viral messages, we infected cells, we immunopurified IFIT2, and then we did RT-qPCR to measure the amount of viral mRNAs. And in all of these RIP experiments, we see that IFIT2 is selectively enriching influenza mRNAs from all 8 genomic segments. In some cases, we're getting 25 to almost 75-fold enhancement of these viral messages. So IFIT2 is binding to viral mRNAs during the context of an infection. It's not binding to viral genomes. So now when we come back in and instead of probing for mRNA, which we see is enriched by IFIT2, if we probe for genomic RNAs, we don't see it there at all. And that's fully consistent with our immunoprecipitation assays, where IFIT2 wasn't co-precipitating with these ribonucleoprotein complexes. Is this a direct interaction? We can make recombinant IFIT2. We can in vitro transcribe cap and polyadenylate influenza mRNAs and ask whether or not they interact. And in these assays, we use gel shifts to measure interaction. A wild-type IFIT2 will shift influenza virus mRNAs, whereas the RNA-binding mutant of IFIT2 is no longer able to do that. So IFIT2 is ripping influenza mRNAs from infected cells, and it's shifting influenza mRNAs in these in vitro assays. Now, what my graduate student Mitch noticed when he was doing these is he looked at the quality of the RNAs we were coming down with, and he saw that we had lots of ribosomal RNAs in our IFIT2 IPs. And most of the time you would dismiss that because ribosomal RNAs come down with everything. But in this case, he saw that it was actually quite specific because the IgG didn't show similar levels of these ribosomal RNAs. And then we probed for ribosomal proteins, and we could see that ribosomal proteins, particularly the large subunit Ripple10, was present in the IFIT2 pull down, but not in the control. Was IFIT2 associating with ribosomes? We could do ribosome pelleting, and we see that during the context of an infection, IFIT2 was pelleting with ribosomes, and that we could disrupt this pelleting by breaking apart ribosomes using EDTA. And when we broke apart the ribosomes using EDTA, IFIT2 would no longer pellet into these cushions. Similarly, we could do polysome profiling and show that when cells are infected, IFIT2 is now reaching deep into the polysome profile. You can use Ripple10 as our marker of ribosomal proteins, and we see IFIT2 trailing way into the deep, heavy polysome gradient. And again, when we break up these profiles using EDTA, IFIT2 now floats to the top along with the ribosomal proteins. It's associating with the ribosomes. Is it enhancing viral gene expression through that interaction? When we look at just virus gene expression, so we can express the polymerase, nucleoprotein, and a reporter, so we're not even infecting cells, we can now assay whether or not IFIT2 is stimulating virus genes. And in this case, that's exactly what we see. Looking in yellow here at a viral gene, so this is firefly luciferase being driven by the influenza polymerase, IFIT2 expression causes an increase, whereas our vanilla control has only marginal effects. And that's our lab strain WSN, smaller effects for the 1918 polymerase, and really large effects for a contemporary strain from New York. That was dependent on the ability of IFIT2 to bind RNA. So once again, we've connected back around RNA binding to functional effects on viral gene expression. And then finally, we wanted to ask in a kind of more rigorous fashion, what is happening to these bound influenza mRNAs that are associating with IFIT2? So we again use polysome profiling from our wild type and our knockout cells to look at the abundance of viral messages that are being translated during the context of an infection. And in our knockout cells, what we see is that in the heavy polysome representing messages that are being efficiently translated, the amount of influenza mRNA decreases by about almost 25 to 30%. So the influenza mRNAs are dropping, whereas a generic cellular control beta-actin is unaffected. And what's the output? What's the consequence? The consequence is that we can get this boost in virus gene expression. And we looked at that in this in a more specific way to see if it was the underlying mRNA sequence that was important. So we took influenza virus, expressed the polymerase, and probed for PB1, one of the subunits. When we have IFIT2, we see a boost in protein production, and that's commensurate then with this observation that we've got more viral messages in the heavy polysomes. But when we express that same protein PB1, but now do it from a codon-optimized template, IFIT2 doesn't matter at all. So this leaves us with an interesting position now, where the IFIT2 is binding to viral mRNAs, and it's the underlying sequence of that mRNA that appears to allow it to enhance the translational efficiency of a viral message. And it brings us back around to this larger conclusion that classical antiviral proteins can be subverted or repurposed or reused during the context of infection into a proviral role. And that's specifically for IFIT2, that in the absence of an infection, IFIT2 is going to bind RNA, and that induces apoptosis. In other infections, such as for VSV, IFIT2 binds to RNA and it blocks those replication events. But in the context of flu, influenza virus has figured out how to hijack IFIT2's activity. We're now using these RNA-protein complexes to enhance virus gene expression, protein production, virion production, and virally mediated cell death. And now what we're thinking about is that that we've evolved to not only evade and exploit the antiviral environment, but we're stealing these cofactors along the way. So then taking it back one step to a larger idea, if you are a cell and you are challenged or you see a pathogen or a virus, this is a highly unusual event to you, and you pull out all the stops and raise all the alarms and establish an antiviral state. You've got pathogen sensors, antipathogen effectors, and you try to block this infection. But of course, viruses are successful. They get around this, and I would argue that acute viruses like flu, the antiviral state doesn't mean anything to them because it's the only state that virus ever sees. There's nothing unusual about it. And if you're going to replicate under those conditions, you have to figure out how to evade detection or evolve resistance. And then I think what our screen has allowed us to tap into a little bit is not only just this idea, but also that we can exploit these canonical antiviral proteins to drive replication. So with that, I just want to recognize the people that did the work. This is my group. It was V, Tran, and Mitch that really drove this project. They go off to JCPenney every year and get a family portrait. It's funny and creepy all at once. They hang in my office kind of looking over me. And then, of course, I mentioned our collaborators along the way, Mike Diamond, Jaco Boone, Martin Schwamla, Adolfo Garcia-Sastre, and then those that contributed reagents. So, thank you.